Okay, so I'd like to introduce Lucy, who's going to tell us a bit about modelling glycans and so forth with Privateer. You're a PhD student working with John Aguirre and Garib, I believe. Okay, so have fun.
Thank you. Thanks. Yeah, I might need to move this down a bit. Hi, I'm Lucy. I'm from the Structural Biology Laboratory and I'm a first year PhD student. Oh, is that all right? Okay, perfect. Sorry. So I work under the supervision of John Aguirre and Garib Murshidov, and today I'm going to tell you about why glycans matter. So firstly, what is a glycan? Well, we get the word glycan or glyco from the Greek word glucose, which means sweet. And in this case, it makes perfect sense because when we think of sweet things, we think of table sugar. And table sugar is a disaccharide, so it's crystallized sucrose, made up of two sugars, fructose and glucose. Sugars have a formal name of carbohydrates, named so because of their carbon backbone and their numerous hydrogen and oxygen atoms, hence hydrate. But we can agree that not all sugars are sweet. For example, cellulose is made of multiple glucose residues bonded together, stabilized by hydrogen bonds. These are examples of free sugars or sugar ligands, but as structural biologists, we're interested in when a sugar molecule is covalently attached to a protein. A common example of this is in antibodies. Here you can see a sugar or glycan chain covalently attached to a protein, and this is known as a glycoprotein or protein glycosylation. This shows a fragment of an antibody, and on the surface you can see a sugar molecule covalently bound. The sugar molecule takes up a small surface of the protein, and actually the protein is made up of mostly non-sugar atoms. But this doesn't stop the sugar from driving the function of the glycoprotein. Protein glycosylation occurs at the co-translational or post-translational level, and this means that unlike proteins, there's no genetic blueprint. This means it's quite a complex and diverse process, and it means that glycosylation profiles between, say, animals and insects can be very different. The most common type of protein glycosylation is N-glycosylation, and this occurs when a sugar molecule is covalently attached to an asparagine residue via the nitrogen atom. It has a strict consensus sequence of asparagine, followed by any residue but proline, followed by a serine or threonine residue. And this tripeptide motif guides N-glycan addition. Glycan addition begins in the endoplasmic reticulum, where a sugar chain made up of mostly mannose residues is covalently attached to a protein. This is then transported to the Golgi, where it travels through the cis and medial Golgi, where glycan branching and extension reactions occur. And it's followed by maturation in the trans Golgi. N-glycans exhibit remarkable diversity, and they can be separated into different categories. We use a common symbol representation for glycans, shown here by the shapes. So it's different shapes and colors represent different sugars. In the top left, you can see an example of a high mannose glycan. These are made up of mostly mannose residues, and they're important for quality control in the ER and protein folding. In the bottom left, you can see an example of a plant glycan. These have high compositional differences from their animal counterparts, including more xylose and fucose residues. In the top right, you can see an example of a complex glycan. These are characterized by being highly branched and contain a high diversity of monosaccharide residues. And in the bottom right, you can see a sialylated complex glycan. And this is where a sialic acid residue has been covalently attached to the terminus of a sugar chain. And this is important for surface glycoproteins. There are other types of protein glycosylation other than N-glycans, and these come in the form of O and C glycans. O-glycosylation occurs when a sugar molecule is covalently attached to a serine or threonine residue via the hydroxyl group. And C-glycosylation occurs when a sugar molecule is covalently attached to a carbon atom in the protein backbone, an example being tryptophan monosylation. These are harder to study because they don't have a consensus sequence, and they're also less frequent in nature. But why do glycans matter? So I'm sure you could all tell me that this is SARS-CoV-2, and on the surface are spike glycoproteins. And it's interesting to see these in action. On the left, you can see the glycans are represented by the common symbol representation. So for example, green spheres are always mannose, blue cubes are always glycnac. This simulation was produced by one of John's collaborators, Elisa Fada, and it shows a glycan chain wiggling into place in a receptor binding domain. And this is important for infection. So it's really interesting that we can see molecules in action because it can tell us something about their behavior. But these techniques rely on the atomic models that they set in motion. So we need to ensure that all atomic models that we're using are correct. So how many atomic models are we dealing with in terms of carbohydrates? In the protein data bank, around 10% of structures contain sugars, and this has been increasing over the past 20 years. They're frequently determined by X-ray crystallography and cryo-EM. This figure is slightly outdated, it's from 2020, so the resolution of cryo-EM has increased since then, but the general trend still holds, which is that as resolution worsens, it becomes more difficult to model glycans into density. And in the absence of expertise, this can lead to guesswork, which leads to modeling errors in sugars. And something that I found over the past few months is that becoming an expert in sugar chemistry is quite difficult. They're not the easiest to work with. So they exist within one of these 5 forms. There's an open chain form in the middle, and then 2 different types of rings, a furanose ring shown on the left, and a pyranose ring shown on the right. There are also 2 different anomeric configurations. The beta anomer is shown on the top, and the alpha anomer is shown on the bottom. And there should be different 3-letter codes for each of these in the protein data bank. To make things more difficult, anomeric configurations can coexist within a crystal structure. On the right, you can see there's density for both the alpha and beta anomer. And also, sugars can have different ring conformations. So in the middle, the sugar is a boat conformer, whereas the other 2 are chair conformers. But what is ring conformation? So ring conformation describes the relative positionings of atoms in a ring. Here you can see a glucose residue. And if we take away the ring substituents, the ring conformation becomes more clear. So this is a 4C1 chair conformation. And sugars like chairs. So chair is the most comfortable conformer for pyranose sugars. And this is because the ring substituents are held in a staggered conformation. There are other high energy conformations that pyranose sugars can adopt, including envelopes, twists, boats. These are high energy because of the angle strain around the ring, and also because the ring substituents are held in an eclipsed manner. This can be defined numerically using the Kremer-Popel algorithm. So out-of-plane deviations of puckered rings can be measured using the Z coordinates of atoms relative to a mean sugar plane. And Q tells us about the overall puckering amplitude or the shape of the ring. On the right, you can see the Kremer-Popel sphere, and this describes all the conformational possibilities of pyranose sugars. So in order to go from a 4C1 chair, which is the lowest energy conformation at the top, to a boat conformation along the middle, the ring has to pass through high energy conformations such as envelopes and half chairs, and this requires a significant energy input. Simulations can show how conformational transitions might work, for example, if a sugar is being distorted by an enzyme. In the middle, you can see the low energy chair conformer, and in order to go to any other conformation, there's a high energy barrier that needs to be overcome. This leads us to an important appropriate assumption that sugars will normally exist in the low energy chair conformation unless they're being distorted by an enzyme. But we've seen that these realities are not well reflected in structures of glycoproteins, as shown by this conformational analysis in the protein data bank. On the right, you can see clusters of sugars in the middle of the graph, and this represents sugars in high energy conformations such as boats and skew boats. As we know, sugars should exist as chairs, so these are the result of modeling errors, interpretation errors, and refinement mistakes. And it's highlighted the need for a carbohydrate-specific method that could be used to produce more chemically feasible models of sugars. And this is what we've been doing to help. So Privateer is a carbohydrate validation software that can scan your structures for unlikely ring conformations, biology-defined glycans, and poorly fitting models. It can be used in combination with standard refinement and model building tools such as RefMac and Coot. Privateers help produce dictionaries and also torsion restraints. And it can help with linkage torsion analysis so that you can compare your sugar link to a validated set of sugars. Privateer has been around for about 8 years, and it's been used in many different relevant contexts, including being used in constructs that informed mRNA vaccine design. It can be used in both CCP4 and CCP-EM. A key implementation in Privateer is the use of glycomics-powered validation. This allows users to compare their glycan composition to glycomics databases mirrored offline. It allows the spotting of inconsistent linkages, such as this beta-6 fucose, which doesn't exist in nature. And it will be suggested that it should be an alpha-6 fucose, and extensions to the glycan chain can be suggested. This is how you run Privateer using CCP4i2. You'll need a PDB file and an MTZ. You can change some of the settings, including the expression system. And you can also ask it to calculate permutations in case you think you have the composition wrong. It doesn't take very long to run. And this is what the results look like. So 2 graphs are produced, a conformational landscape based on the cremopopal sphere, and RSCC versus B factor. Here you can see there's one conformational outlier, and it's listed in the results table as an envelope. You can also look at 2D descriptions of the glycans using the common symbol representation that we mentioned earlier. This shows links to the glycomics databases, and also hovering over any of the sugars or links will tell you more information about them. This is another example. So this one has a lot more conformational outliers. It's listed in the results table as envelopes, U-boats, half chairs. And if we look at the 2D description, you can see that this has not been identified in the glycomics database. And actually, it will suggest closest permutations so that you can correct your glycan composition to one that does exist within the glycomics database. This is how you run Privateer using CCP-EM. You'll need a PDB file and a map. You can also change the resolution. Again, it doesn't take very long to run. And the results look very similar to CCP4i2. So there are 2 plots generated, a conformational landscape and RSCC versus B factor. You can look at the 2D descriptions, including links to the glycomics databases. And again, hovering over any of the sugars or links will give you more information about them. You can also see a more in detailed results table, which gives you a list of all the sugars in your model. I'll now talk you through how to validate a glycan using both Privateer and Coot. I'm using this example structure 3QVP, and I ran it through Privateer using CCP4i2. This is the output I got, so you can see we have one conformational outlier, and it's listed in the results table as a skew boat. I then went into Coot, and the issue with this model was that the solvent was overfit, and this was pushing sugars out of electron density and into high energy conformations. So the first thing I did was got rid of the solvent and reran this through RefMac. We then took that output and looked at it in Coot. Coot has a carbohydrate-specific tool that can be used, which is called Glyco. And I use it to do 2 things. So firstly, I use the refine tree tool, which changes the high energy conformation to a low energy conformation. And I also used it to add in more sugars based on the electron density that had now become free after we'd removed the overfit solvent. So the first thing was to use the refined tree tool that changes it to a chair conformation, which is what we want. We want the low energy conformation. Then we extend the glycan chain based on electron density that's become free due to removing the overfit solvent. And there's also some electron density next to this beta mannose residue. And again, we can add in a sugar. We then took this structure and rerun it through RefMac, and it produced the structure on the right. So we started with the structure on the left. This has a relatively simple glycan chain. It has sugars in high energy conformations that are being pushed out of electron density due to an overfit solvent. And we've produced the structure on the right. This has a more detailed glycan chain that's been extended based on appropriate electron density, and all of the sugars are in the low energy conformation. When we rerun this structure through Privateer, we get no conformational outliers. To try and reduce the chance of producing distorted models, we've generated standard dictionaries using both Privateer and AceDrug. This incorporates a set of validated unimodal torsion restraints that have been tested extensively and can be used in refinement like any other set of restraints. Torsion restraints match the lowest energy conformer in the dictionary, and this is what will be placed in the model building tool. And in RefMac, you can enable the torsion restraints separately from the rest of the sets. We found that this has reduced conformational outliers for N-glycans. So you can see when we use the old dictionaries, we get this cluster of sugars in the middle of the graph representing sugars in high energy conformations such as boats and skew boats. But when we use the new dictionaries and torsion restraints, we reduce the number of conformational outliers. So that was for N-glycans, but what about for sugar ligands? You can see a similar trend that's shown on the right. But importantly, for sugars in high energy conformations that are well determined, as shown here, these have not been changed. And interestingly, the torsion restraints reduce R gap, which reduces overfitting in your model. There's been an effort to improve restraint consistency across software, including Privateer, Pyrogen, Grade, AceDrug, and Phoenix Elbow. And this was done by getting in touch with developers. This was with the aim to improve results consistency, which has been achieved, as you can see on the right. The new version of Privateer will aim to get tryptophan monosylation right. This is a type of C-glycosylation. The mannose residue is found nearly always distorted in examples of C-mannosylation in the protein data bank. And this isn't helped by the fact that the diagram in our reference textbook was wrong. So it showed a beta link rather than the expected alpha one. This has since been updated. It now shows the alpha link with the expected 1C4 inverted chair, which reduces clashes between the ring substituents. This is an example of a paper that's used Privateer to generate external restraints for tryptophan monosylation, and it correctly placed the mannose residue in a 1C4 inverted chair with an alpha link. Privateer will help both detect and fix issues with tryptophan monosylation using two methods, firstly through standard dictionaries and also through external restraints. Privateer can also help with analysis of linkage torsions. This allows you to compare your sugar link to a validated set of sugars. Privateer can produce these torsional plots shown on the right for the asparagine NAG linkage. Here you can see there are two common torsion pairs, and these are modeled on the left. Privateer can plot your sugar link on this graph, and it will inform you whether your sugar links are torsional outliers. We performed some outlier analysis ourselves, and we found that there are two reasons for outliers. Firstly, they're the result of modeling errors, or they're chemically interesting. So on the left, you can see an example that's the result of a modeling error. So there's a high energy conformation that's pushing the sugar link into a standout conformation, which is resulting in torsional outliers. Whereas on the right, this is an example that's chemically interesting. The sugar link fits well within the electron density. They're low energy conformers, and it's actually backed up with interactions with the protein. And it suggests that this protein is distorting this sugar link, which is causing the torsional outliers. This is work that I've been heavily involved with. So we've implemented algorithms to detect XH pi interactions, which are weak hydrogen bond-like interactions that form between proteins and sugars. This is based on a set of geometric criteria, and it has implications for both glycan stability and protein sugar ligand interactions. And we'll be doing more work on this in the future. Our most recent development is the Privateer web app. The URL is at the bottom. We'd like people to test it and give us feedback. This allows you to access a browser version of Privateer. You can use your own structures, or you can fetch straight from the protein data bank. The initial results output is a list of glycan chains in your structure, and you can click into them to get more information. There's a window that allows you to visualize the structure with Moorhen, and clicking on the sugars in the SNFG diagram, so the 2D description on the left, will take you to that exact sugar in Moorhen. You can also see the torsional plots with your sugar link plotted on it, and you can actually move around different sections of the page to put it in a position that makes most sense to you. So I hope I've managed to convince you that glycans matter, and we'll recap some key points. So firstly, in terms of glycan composition, we need to make sure that the glycans you've modeled match biosynthetic pathways, and the way that we do this is using glycomics databases. In terms of ring conformations, high energy puckers are almost never true. They're mostly the result of modeling errors and need to be corrected. And finally, in terms of linkage analysis, modeling errors can force your links into surprising conformations, but not all standard conformations are incorrect, and it could be the result of interactions with your protein. I'd like to take this time to thank the Glycogens team. As Dorothy said earlier, I'm just the presenter, and all of these people have contributed to this work. I'd also like to thank John, who we're all so lucky to work under the supervision of. And I'd also like to acknowledge all of John's collaborators and funders, and I'm happy to take any questions.
Thank you very much. So we have plenty of time for questions, but anybody has them? I think Dave gets in first because he's in the front.
Thanks. That was a really nice and clear presentation on how to spot errors and correct errors. What do you do in terms of the initial assignment of those carbohydrates, and what do you deal with where the density phase disappears partway through what your expected glycan chain? Because obviously these are normally on the surface of the protein, and when they're going off into space, they're less well ordered.
In terms of modeling, Coot has a glycan builder that's useful. It has different modes so that you can use, I think it's, I'm not sure about the name of it, but you can add in whole glycan chains based on whether it's a high mannose glycan or a complex glycan and that kind of thing. And then kind of with density as shown here, I would model until the density disappears. I don't know if John, you wanted to add anything? No? Okay.
I guess if it's an unnatural termination,
is
there a way of flagging that? Well, if you're only modeling the first two carbohydrates, the first two sugars, and you're expecting these five, how do you flag what that is? And then probably what's in the system is the full sugar, but you only modeling part of it. So in a protein, you've got a full sequence. So on a protein, you know what the sequence of the protein is you put in. For here, we don't know what the glycosylation actually is unless we've measured it through mass spec glycans mapping.
Thank you, David. So at the moment, there is no way to annotate that, but we hope that using the MMC format, we can add something, because that's a really interesting point.
There are some questions on Slack. So Maria asked, does anyone know how good the glycan data in the CSD/COD is? For example, the recent Rosetta fold all atom that can co-fold glycans as well as other ligands with the protein was trained on PDB plus CSD. And she wonders whether it's good enough to predict glycans well.
That's a really interesting question, but I think it should be given to somebody more qualified to answer.
Yeah, John Aguirre, University of York. I think the amount of data and quality of data that is in the PDB is not great for doing things like deep learning and so on. So whatever predictions are made based on those data, but might probably not work really well. We've tried doing that, and well, we hit a wall with the lack of data. I don't know if that answered the question or.
Another one from Slack from Rob. I'm curious, how well would Privateer handle intermediate states in soaks? It's unclear to me if it would treat them as outliers.
John, do you want to take this one?
I'm not sure what the question refers to. I'm guessing this has to do with partially occupied sites. So Privateer will look for conformation A, conformation B, and whatever you got in the structure would treat them as separate entities.
Is it not like a transition state mimic that might?
Is that what the question is about?
That's
Like,
how I interpret it. Is like maybe the transition state mimic is in an odd conformation.
right.
Perhaps it's
Yes.
a question for a pub.
I think, yes, but this sounds like a bar discussion. You guys can pick it up later.
Okay.
Cool, thank you. Okay, if we could all thank Lucy once more.